Hello. In this video, I'll be discussing the Catabolite Activator Protein or CAP. It exists as a dimer of two identical subunits, each with 209 residues. Here's the secondary structure. It has alpha helices as well as beta sheets, all connected via loops. Each subunit has one DNA binding unit and another domain to bind cyclic adenosine monophosphate or CAMP. Now, the binding domain includes a helix turn helix motif, which, by the way, is a type of motif that is conserved across plenty of other DNA binding proteins. It's simply an alpha helix connected to another alpha helix by a short stretch of amino acids. Now, in the absence of CAMP, this DNA binding motif is inaccessible. But when CAMP is present, it modifies CAP allosterically and the motif becomes accessible, which binds to the DNA's major grooves. This enables CAP to also bend the DNA significantly. It closes the major groove and widens the opposing minor grooves, making the DNA more accessible to the RNA polymerase that it recruits. CAP has many positively charged residues like histidine and lysine, which interact with the negatively charged DNA phosphates and stabilize this bend. Now, this was all about the structure. CAP also serves as a classical model system for studying transcriptional activation. One such very well studied example is CAP's involvement in regulating the lac operon. So the lac operon is turned on or off majorly by two switches, the lac repressor and CAP. So what happens is when glucose levels are low, CAMP levels increase and CAMP binds to CAP, which enables it to bind to the CAP site on DNA. This then recruits the RNA polymerase and this initiates transcription. Meanwhile, in the absence of lactose, lac repressor is bound to its operator site. Now, this acts as a roadblock, preventing RNA polymerase from transcribing the lac operon. But when lactose is present, it converts to allolactose, which binds to the lac repressor, causing it to release the operator site and allow transcription. So overall, this regulation system can be represented by this logic circuit, and as you can see, this system ensures, this very intricate, smart, organized system ensures that the lac operon is switched on only when lactose levels are high and glucose levels are low simultaneously. So this was all about the structure and one of the very well studied, interesting functions of CAP. And this is all that I could cover in under 3 minutes. These are the papers that I referred to, and all photographs that have been used have been obtained from their respective PDB files using Pymon. And I hope you enjoyed the presentation. Thank you very much.